Now students, after the counter current mechanism, now we are to the, you can say the third part of the urine formation, as we have discussed the very first part as the ultrafiltration. The second part was the reabsorption. And now we have the third part, which is called as the tubular secretion. Now, this tubular secretion is into the lumen of the, you can say the tubules. Now the nephron, it has several distinct systems that secrete substances by transporting them from the plasma into the tubular lumen. Now most investigated kind of the ions or the systems of secretion, these are of potassium, hydrogen, ammonia, organic acids, and the organic bases. Now, the number of the secretory mechanisms and the transport molecules, they must be limited in some amount. The nephron is capable of secreting drugs and toxins which are taken in by the humans into the urine. So as well as some of the endogenous naturally occurring molecules or which are being formed as a part of the metabolism. So they are also secreted out in the urine. So how the nephron is able to recognize and transport all these diverse kind of substances. So this is a very important thing. Now how the kidney knows that this particular substance has to be removed in the urine from the blood. Now this is the important thing. Now the answer to this particular question, it is actually present in the vertebrate liver. So this is the important organ which is modifying many of the molecules which are present in the blood. So when the liver, it modifies a particular substance, they can react with the transport systems which are located in the wall of the nephron. So suppose you have a particular substance which has to be removed out from the body through the urine. So this particular substance, first of all, the liver, it is modifying this particular substance, and suppose we have a particular modification just like this. So there is a particular part of the compound or a particular chemical substance that is added to this molecule, and now this is a, you can say, a modified kind of a molecule, and now it will be reacting with the wall of the nephron, and the nephron will take this particular thing into the tubule and it will be given out in the urine. So this modification which is going on in the liver, it is very, very important as per the excretion of these kinds of substances or the toxins or the drugs it is concerned. Now if we talk further about the secretory mechanisms, so they are very, very important because they remove many dangerous or potentially harmful substances from the blood out in the urine. Now in the liver, many of these substances along with the normal metabolites, they are conjugated with glucuronic acid or the sulfate. So these are the two substances with which the liver is modifying these molecules to be given out of the body. Now the first thing is the glucuronic acid. The second is the sulfate. Now both these classes of the conjugated molecules, they are actively transported by the urinary system. So whenever the kidney, it is taking these conjugated molecules, those will be excreted actively into the urine. So they are just like, they are treated like VIPs. So if they are modified by the liver, They are treated as VIPs and they are actively given out in the urine. Now, once deposited by the transport machinery in the lumen of the nephron, so once the modified substances, when they go to the lumen of the nephron, they cannot readily diffuse back across the wall of the nephron. So once they are actively transported into the lumen of the tubule, once they are secreted into the lumen, and they will not be coming back into the blood. So this is a one-way kind of a traffic. So they are taken as I've said, these are taken as VIPs, and even VIPs, they cannot come back into the blood. So this is a kind of mechanism. So these kind of substances, they are excreted in the urine. So this is the mechanism that follows because of the liver. So students, that was a part of the secretion. Now we have some concepts in the secretion part. The first concept is the pH regulation by the kidney. So this is a very, very important thing. And if we go for the secretion of potassium ions or the secretion of some other ions too, will be taking up later on. So they are all regulating the pH of the fluids of the body. So the kidney, it is very much helpful in regulating the pH of the body fluids. Now students, this is the first diagram which is showing you the pH regulation, and this is by the reabsorption of the filtered bicarbonate ions and giving out the sodium ions also. So here what is being depicted is that you have the bicarbonate ions in the tubular lumen over here. So these are the filtered out bicarbonate ions and they are going out into the urine. So first of all, I will give you a concept that this is the tubular lumen. This is the lumen of the tubule, and if something is present here, it will be going out into the urine. Now this is a renal tubular cell. So that means it is a particular cell of the proximal convoluted tubule or the distal convoluted tubule. So we have a peritubular plasma space over here. So this is the in-between space of the cells. So these are the three spaces in which these ions, they are moving across. So here we have the bicarbonate ions in the tubular lumen, and what is happening that here is the sodium and hydrogen ion pump present. So this pump is pumping out the hydrogen ions. So as you can very well see, in the lieu of one sodium ion intake. So in the tubular lumen, what we had is the sodium bicarbonate. So here you can very well see that we have the sodium bicarbonate which bifurcates into sodium ion and bicarbonate ion. Now from here, the sodium ions, they are taken in, and the hydrogen ions, they are going out. So this is a kind of pH regulation. So it is making some acidity, it is giving some acidity to the tubular lumen fluid. As the hydrogen ions, they are passing out. So pH is being lowered. It is made to some extent more acidic in nature. So when the hydrogen ion, it comes out, it reacts with the bicarbonate ion and it forms the carbonic acid in the tubular lumen. Now this carbonic acid, it can be given out or it dissociates into water and carbon dioxide, as you can very well see. Now these two things, they are traveling into the cell. From here, the carbon dioxide can leave into the peritubular plasma, Otherwise, both can recombine to form the carbonic acid with the help of the carbonic anhydrase enzyme. Now, this is again bifurcating into the hydrogen ions and the bicarbonate ions. So, what actually has happened in this particular mechanism? So, here you can see that what is going into the peritubular plasma. So, this is the most important thing. If you see this particular part, this is an active sodium potassium pump. Now it is pumping the sodium ions into the peritubular plasma. So ultimately, if I go back, if I wash everything, what is actually given in and out? It is, if we go for the sodium ions, they are coming first of all in, they are into the peritubular plasma, they are reabsorbed. If I talk about the bicarbonate ions, they are also ultimately they are going into the peritubular plasma. So they are reabsorbed. So students, via going into this part and then here, then coming back and then up to here, the bicarbonate ions, they are reabsorbed. So if we are taking the bicarbonate ions and giving some of the hydrogen ion out, so this is a kind of a balance of pH. This is a kind of buffer system that is happening in the renal tubular cells. So this is a kind of regulation of the pH. Now, if we go for this particular diagram, now here what is happening that it is showing the excretion of the acid or the hydrogen ions out in the urine. So how this happens and how it is, you can say, occurring, that we have got sodium hypophosphate. So it is Na2HPO4. So here the Na or the sodium ions, they have, you can say, dissociated, and these are taken in, as we have seen in the previous diagram also. So they are again into the peritubular plasma. Hydrogen ions, they are coming out. Now what happens that these hydrogen ions, they react with the sodium hypophosphate ions. So what they are making is Na plus, it is coming out of the compound. If we go from here to here, what we have is the hypophosphate ions, and these are given out in the final urine. So this is a kind of thing which is going on to give out the extra hydrogen ions. So if I summarize by going back, if I wash everything, now if we talk about the hydrogen ions. So here the excretion of the hydrogen ions is being done in the form of hypophosphate, you can say, acid. So here the hydrogen ions, they are being given in the tubular lumen, and these hydrogen ions, they are reacting with the compounds, and here we have again the hydrogen as H2, and this hydrogen is going out into the final urine. So this is a kind of mechanism of excretion of extra H ions if you have in the tubular lumen into the urine. So again, this is a kind of pH regulation. Now students, if we go by this particular diagram, here it has been shown that the hydrogen ions, they are given out, they are excreted in the urine as the ammonium ions. So what is happening over here, that if we have some amino acids which are giving out the ammonia, here it has been written as the glutamine and the amino acids. So they are giving you ammonia. So this is NH3. Now this ammonia can go for two pathways. So first is into the peritubular plasma, or it can go into the tubular lumen. So here also the hydrogen ions, they are reacting with this ammonia to form the ammonium ions. So here also in the tubular lumen, the ammonium ions are formed by the reaction with the hydrogen ions, which are given out by the renal tubular cells. So these ammonium ions, which are formed in the tubular lumen, they are given out in the final urine. So this is again a kind of pH regulation by excreting the ammonium ions. So students, that was the part of the kidney that you can get a small question on, that how the kidney is regulating the pH. So you can give these three diagrams and you can explain that how the bicarbonate buffer system is occurring in the kidney cells and how the hydrogen ions, they are given out in the form of H2PO4 and ammonium ions.